Okay, here's my mini lecture on some of what's in Chapter 6. Since most of us humans don't have superpowers, we can't hear everything, see everything, or sense everything around us. The absolute threshold refers to the smallest intensity stimulus we can sense, 50% of the time. Most of the research is focused on our sense of hearing, but this applies to every sense. In lab tests, people can detect a whisper up to a specific decibel level 50% of the time. That's their absolute threshold. People vary in their abilities, but there are normal ranges for absolute thresholds. Our abilities can be impacted by background noises, which can prevent us from zeroing in on one voice or sound. Or if we're trying to see a very specific leaf in a jungle, that will be tougher with all of the other leaves catching our eyes. We are also able to detect the differences when stimuli change. If somebody first whispers to you and then starts yelling loudly, you can easily tell the difference between the whisper and the yell. But what if they gradually increase their volume? At what point would you notice that it's now louder? The just noticeable difference, also called the difference threshold, refers to the minimum difference between two stimuli that a person can detect 50% of the time. If I were to show you a handful of 5 marbles, you'd easily be able to tell if I added one more marble, making 6, right? Have you ever heard of stores running contests with a big jar of marbles trying to see who can guess the number? Well, if I added one more marble to one of those jars of something like 950 marbles, would you be able to tell? Most of us would not. Weber's law explains this by stating that in order for us to perceive a difference, the two stimuli must differ by a constant percentage or ratio. You'd have to add a lot more than just one marble to that jar of 950 to notice the difference. Our senses can be pretty amazing though. If we're in a dark cave and there's a single candle lit at the other end of the cave, you'll notice when that one candle goes out. That one candle could even be miles away. But then we won't notice if there are millions of lit candles and only one of them goes out. Did you see this example in the book with the font size gradually increasing? The average person doesn't notice the increase until line 6, 7, or 8. Human beings can't see every wavelength. Highlighted here is the visible light we can see with UV rays and infrared rays on either side, along with several others, which we can't see. In this graphic, there's a candle in this eye's line of sight. But why is the candle upside down back here in the fovea? Our eyes send sensory messages that are upside down to the retina, but then through the process of perception, as the sensory messages are interpreted and perceived by the brain, images are understood as being right side up. The eye is curved, which bends light and makes things appear upside down. We want light to bend because that allows us to focus. So what happens when our eyes first sense light energy? How and where does that sensory message travel? First, it hits the cornea on the outside of our eye. The cornea protects our eyes and also bends light, at least somewhat, so that it can begin to be focused. Light next passes through our iris, which is our unique colored area. I say unique because, like fingerprints, it's our iris that makes our eyes unique enough to be identified by software programs providing security. The iris controls the muscles that flex or release so that our pupil, the black part in the center of the iris, either dilates, which means it gets bigger to allow more light in, or contracts, it gets smaller to allow less light in. When it's dark, our pupil will be dilated, and vice versa when it's very bright, then it's contracted. Our emotions and medications can also result in the dilation of our pupil. When light hits the lens further focuses the image, depending on the curvature and thickness of the lens. The image is then projected back onto the retina, which is in the back of the eye, and that's when the image gets turned upside down. The retina converts or transduces the light. Transduces means that process of conversion of sensory input to something that the brain can perceive. So after the retina transduces the light, which is in the form of the electromagnetic energy, into electrical messages that can travel to the brain, that passes via the optic nerve. Rods and cones are within the retina. These are the receptors that allow us to see in the dark and out of the sides of our eyes, the rods, and to see different hues or variations in color, the cones. Cones allow us to see fine details, and rods allow us to adjust to changes in the brightness of our lighting. On this slide, I've shown you the progression of light traveling through your eye, starting first with the cornea, all the way down or back to the retina. Your optic nerve carries messages from your eye to your brain to the thalamus. But where the optic nerve first leaves the eye, there's a blind spot where there are no receptors to receive incoming visual data. You just can't see anything in that spot. In your textbook, there's an exercise where you can stare at a dot in between two images of cars, close one eye, and move the page toward your face until you can't see one of the cars, the one that drops off in the blind spot. Each of your eyes sees more than what's immediately in front of it. They each have a visual field. Once images are processed by the eye, neural impulses get to the opposite hemisphere in the brain. So what your right eye processes gets sent to the left hemisphere in the brain, and vice versa. In our brain's occipital lobe's visual cortex, there are specialized neurons that respond to specific shapes or features. There are also specific neurons in the temporal lobe, in the association area's gray matter, that can recognize faces. So every time we see a face, those specific neurons in that specific place of our temporal lobe will light up. But just because these neurons specialize, that doesn't mean they work alone. There are a lot of other places in the brain that might be activated when you see a face, maybe someone you recognize. In that case, areas responsible for your memory will also be activated. Maybe your limbic system will light up if you love that person. And your experience or your expectations definitely play a role in what you perceive. We tend to see what we think we're going to see. I bet most of y'all will be able to read this because of that tendency to fill in gaps and see what we expect. Here, we're also able to use context, based on our years of reading, to guess at what the next word will be. This shows how after we become adept at reading, we no longer have to analyze each and every letter of a word to know what that word is. For a mini recap and a bit more detail, when we see something, like the scene depicted here on this slide, the image gets sent back to the retina, where the rods and cones receive the light energy, and then a chemical reaction takes place. First, cells known as bipolar cells are activated, and then the ganglion cells are activated. The axons of the ganglion cells are what twist together to form the optic nerve, which carries neural impulses to the brain's thalamus. What does the thalamus do? It's the sensory router, receiving and routing all sensory information except smell. The brain then begins its work to make sense of what the eyes have sensed. Those specialized feature detection cells recognize specific shapes or patterns like faces, and to truly make sense of a scene, the brain will tap into memories and prior experiences. When we recognize what we're seeing and understand it, that's when perception happens. But remember, sometimes we fill in a lot of blanks in order to reach some understanding. Sometimes we see things that aren't quite there. Now let's talk about our perception of color. First, why do I call it the perception of color versus the sensing of color, or even the reception of color? Is an apple actually red? Light has no color. It's our brains that perceive color because of how the light reflects off of an object. We see a red apple because it's the red light that has been rejected by the apple and is thus reflecting off of it. Our senses only pick up on signals, but it's the perception that allows us to actually understand those signals. The Trichromatic Theory, or in the textbook, the Young-Helmholtz theory, suggests that we have three different types of cones in our retinas. One type of cone is stimulated by blue colors, another is stimulated by greens, and a third is stimulated by reds. While there isn't a cone that specializes in violet or yellow, when a combination of the three types of cones are stimulated, we see colors that vary from the standard blue, green, and red, like violet or yellow. For yellow, it'd be the red sensitive and the green sensitive cones that both activated. The degree to which each of these three types of cones is activated will result in a particular hue. If both the blue sensitive and the green sensitive cones are activated at about the same degree, then we'll see a color like teal. If only the green sensitive cones are activated, it'll be a true green. There's an activity in the textbook that asks you to stare at the image you see for a full minute. Then when you shift your gaze to a plain white page, you should see different colors than the ones you started with. That's called an after image. In the book, you first saw green, yellow, and black, but in the after image, you'll see red, white, and blue. After images can happen because of the opponent process theory, which says that there are three retinal processes: red-green, yellow-blue, and white-black. Some neurons will turn on for red, but when they do, they're turned off for green. They can only send one message at a time. So when your eye is processing red, green is blocked from traveling down the optic nerve via that particular neuron. The colors red and green are opponents. The same goes for the other processes. You can only send a message down a single neuron for yellow or blue, and for white or black. By staring at the image in the textbook, you wore out the neurons for the colors you perceived, the green, yellow, and black. And that's why the messages for the opposite colors were seen in the after image when you had a blank page. Your neurons got sick of green, yellow, and black, and you saw the opposites, red, white, and blue. So theorists today believe that color vision happens first according to the Trichromatic Theory, with specific cones activating in the retina, and then processing takes place according to the opponent process theory, with the eye sending messages to the brain via 3 opponent tracks. One more thing, very few people are truly colorblind, although there is red-green color blindness. A lot of people just don't have well-functioning cones. I have a lot of trouble distinguishing hues. I can see red and green, and I know the difference between those two and other colors, but I can't always tell the difference between black and navy, between orange, pink, and red. Some people, again, I'm one such person, poor me, have poor peripheral vision or night blindness, which are problems with the rods. After vision, it's hearing or audition that gets the next largest amount of brain space. As we talk, keep in mind the anatomy of the ear, which you see here. And what part of the brain is primarily responsible for processing what we hear? The temporal lobe. Sound first enters by way of our outer ear, which is mechanical. Almost like setting off dominoes, sound waves enter the outer ear and then a chain reaction begins as the sound gets funneled through the auditory canal to the eardrum, which then vibrates. Next, in the middle ear, the sound is amplified as it's sent to the inner ear's cochlea. The middle ear is made up of 3 itty-bitty bones, the hammer, anvil, and stirrup. They pick up on the vibrations and pass them along to the inner ear's cochlea. It's within the inner ear that the sound energy is transduced or converted into electrical impulses that can travel to the brain. When the vibrations first hit the cochlea's outer membrane, the oval window, vibrates and shakes around the fluid that's inside the cochlea. Then hair cells that are in the basilar membrane, which is also in the cochlea, start moving and bending as the sound waves move by. And that sets off the process that transduces the sensation into neural messages that get sent to the thalamus in the brain. There are 2 primary theories regarding hearing. The place theory suggests that we hear different pitches or tones because of where in the cochlea's basilar membrane the sound is activated. A high pitch is typically processed near the beginning of the membrane. Low pitches are often near the end, but aren't always exactly in a set location. So place theory does a good job explaining how we hear high frequency or high pitched sounds, but doesn't quite explain low pitched sounds. Frequency theory proposes that the entire basilar membrane vibrates with incoming sound. Frequency is determined by the number of neurons that fire. Lots start firing for higher frequencies, while fewer fire for lower. Critics of this theory point out that a single neuron can't possibly fire at a rate that would result in our ability to perceive the highest pitches that we can actually hear, and they wonder if enough neurons could work together to result in perception of these highest frequencies. That would be called the Volley Principle and is discussed in your book. The book makes it sound like all researchers agree, but there's still some debate about whether or not these 2 theories adequately explain how we hear pitches. So frequency theory does well at explaining low-pitched sounds but not high, and place theory does well explaining high-pitched sounds but not low. Substance P is a peptide and is released in large amounts when we're under stress or when we're presented with some sort of unpleasant stimulus. Some theorists believe it's involved in our perception of pain and that it's released after an injury and resulting inflammation occur. The gate control theory of pain proposes that within the spinal cord there's a sort of gate that either opens to receive pain signals or closes, and then we do not feel pain. You can stimulate the gate to close, alleviating pain, and that's precisely what acupuncture does. Acupuncture, which is sticking needles into specific areas of the body, and also certain drugs, can stimulate this gate to close, and then the pain goes away, or rather, our perception of that pain goes away, at least for a brief period of time. It's important to remember that pain is heavily influenced by whether or not we attend to it. When I fell off a cliff and broke my back, I was worried about a lost friend and had my adrenaline pumping. When I climbed back up, I then kept running down the mountain I just slipped off of. It wasn't until much later that I realized I had a significant injury. When I went to physical therapy and was around other people in pain, I tended to hurt more. You notice when others are in pain and you can mirror their pain. Have you ever heard someone describing a horrible injury and it made your old injury ache? Placebos, or fake drugs and interventions, can help ease pain by prompting the body to release endorphins, the body's natural painkillers. Simply distracting yourself from the pain by not thinking about it and getting busy with something else can also help. What is hypnosis? How does it work? Do you lose power over your mind? Do you enter into a different state of consciousness? Is it a natural thing that happens just because, as social beings, we can allow ourselves to be influenced by what others tell us? Or does it actually create a different level of consciousness? And how can it help us? Here's the definition directly from our book. Hypnotism relies on the participants' willingness to participate and on how suggestible they are. How open are they to following along with what the hypnotist leads them through? There's controversy about whether hypnotism is simply two people agreeing to play out this scenario or if it truly produces an altered state of consciousness. Proponents of the theory of hypnosis as socially influenced behavior believe that all of this is perfectly natural. People will follow leaders, especially if they like them. Hypnotized people will go along with it, letting the hypnotist guide their thoughts. But if they don't like the hypnotist or the hypnotist makes fun of them, suddenly they're no longer hypnotized. So researchers following this theory think that all of this is just a normal, natural phenomenon that happens because we're social creatures. We listen to each other and we can be influenced by one another, but only to a point. Other researchers study the possibility that hypnosis goes a little further than normal social interactions and actually creates dissociation, which is basically split consciousness. It may be that when we're hypnotized, we aren't able to pay attention to other things that we'd normally attend to. Instead, we focus on whatever message the hypnotist is telling us. It's a lot like selective attention, which we've studied earlier, but in this case, it's split between unconscious and conscious attention. In selective attention and inattention, we're consciously and actively ignoring some things, but if we shifted our focus, we'd be aware of them. They'd be part of our conscious awareness. With dissociation, we actually lose the ability to be aware of the things going on in our unconscious minds. Our sense of taste, or gustation, allows us to sense sweet, salty, sour, bitter, and now we've added umami, which is meaty or savory. We have specific receptors, mainly on our tongue, but also in our mouths and down our throats, that receive one of these specific tastes, and every flavor is a combination of the messages sent from these different taste receptors working together. Taste involves a chemical reaction, like olfaction, which is our sense of smell. Our taste receptors are in our taste buds, which again, most people associate with our tongues, but we have taste buds going down our throats too. Most of our taste buds are on the tongue, but not all. We have about 10,000 taste buds. If you burn your mouth, you'll be okay in a few days because every 10 days they're replaced. This regrowth slows down as we age, though. That, along with our emotions, our expectations of what we're about to taste, and genetic differences, result in each person having a different taste experience and different preferences. Some people are super tasters, and they can easily identify ingredients and subtle flavors. Others don't have much sense of taste at all. They're at risk for becoming overweight and even obese because they eat sweet and fatty foods to get some flavor. I watched a show called iZombie. Zombies are not super tasters, since they have to put hot peppers on everything. Olfaction is the scientific term for the sense of smell. When we inhale or breathe even a single molecule in, that sets off a chemical process. An olfactory cell responds to just one specific odor molecule. Multiple olfactory cells work together to arrive at a distinct odor message. The book compared this to using letters in the alphabet to form unique words. Our olfactory cells work together to result in the recognition of a specific odor, and we're able to tell the difference between about 1 trillion distinct odors. We remember things tied to smells best, in large part because of the physical proximity of these brain centers. Olfactory processing takes place right near the amygdala and other memory centers. I originally learned that we had 5 senses, and a 6th sense referred to a supernatural ability to see ghosts. I watched that movie The Sixth Sense with Bruce Willis. But today, psychologists believe that all of us have six senses, with the sixth being kinesthesis, or the sense of body position and movement. Our inner ears play a huge role in this sixth sense. In your inner ear, you have semicircular canals that connect to the cochlea by way of the vestibular sacs. Within the semicircular canals, fluid sloshes around when we move our heads around. Depending on how this fluid moves, which is a mechanical process, our brain understands how we are moving and positioned in space. If people have an inner ear infection, they can often suffer from vertigo or extreme dizziness because their inner ears aren't functioning properly and their brains are confused by their motion.